Good morning, everybody. This is another opportunity to meet again, once again, for bioinformatics. Today is going to be very, very um, long, so to say, because we are going to. I'm going to go through lecture for week 2, week 3, week 4, and probably maybe week 5. Don't forget what I've said so far, that you are expected to do everything you ought to do so as to make sure that you get ready for what is coming up, and that is nothing but for your mid semester. Okay. Now, um, today I'm going to last week and week before we look at bioinformatics. What is bioinformatics? Where we made mention of where science meets computer. Then thereafter, we also look at DNA, genes, and computers, from the cell to computer. Today, we will continue from week 3. That's going to be microbes, that is microorganism, and data. Are there point of intersection between microorganisms and data? And if there are, what are we expected to know about microorganism and data? What are the synergy between the two? Um, so we'll look at that. And that's going to make us focus on bacteria, viruses, and what their digital fingerprints could be. Don't forget that if you mention anything like fingerprints, that is to tell you that DNA is involved, because DNA is the genetic material of every organism. So thereafter, we will also move to online biology databases. So I'm going to introduce you to the world's largest biology libraries. We will look at that. Then we will also guide our tour to database libraries like NCBI, the one that is also used by WHO. So then, in the before we got so organized, before we started counting week 1 to week 3 and so on, I've actually taught you the practical class of blast. That is talking about what is so important about blast. That is basic local alignment sequence tool. And apart from that, I remember that time that I also made you to know what are the significance of blast. So what do we mean by blast? Multiple sequence alignments, and what we have to do with pairwise alignment. So without wasting our time, um, let's go straight to starting today's lecture. And just like I told you, as the lecture will be going on, I want you to concentrate also on the comment section, because I will be giving you some questions, and that's why I said you should get ready with your writing materials and so to say. Um, yes, like I remember I gave you some tasks, role play activities, stepping into science. Nobody has ever done that. I know that. Also the group assignments which I gave you group assignments that you should divide yourself into 5 groups. Number 1, I gave you that the first group should work on disease and data, focusing on malaria. Group 2 should focus on disease and data, focusing on Lassa fever. Group 3 should do something about disease and data, still disease and data. They should focus on cholera. Then group 4 should talk about bioinformatics career research. So they should focus on that. Then again, I also gave you some deliverables in each of these groups. For example, group 4, the only deliverable is a written list of 3 opportunities with brief comments about them that should be submitted. Group 5 should talk about global versus African science, and the deliverables should be 3 written arguments, 1 paragraph each, that should be submitted. Having said that, we move to the next one. I remember I gave you sometimes ago, maybe 2-3 weeks ago, we talked about the DNA, genes, and computer, and I told you what DNA is all about. That is, it is the molecule of life. I also taught you about what genes are. That is, genes are just some segments of the DNA. They are just subsegments of the DNA. Then, apart from that, I also made mention of the Human Genome Project, which I said it started in 1990, then down to the year 2003. And I said that was when the whole world came back with whole genome sequencing of human genome. And for the first time, human genome is made up of more than 3 billion base pair that is going to fill up to 200 large textbooks, and that is the reason why we actually need a computer to do it, and that is when they introduce what bioinformatics is. Then I also made mention of genes within the genome. So at this level, you should have known the differences between gene and genome. I said a gene is a specific segment on DNA, whereas the combination of all these genes within an organism is known as the genome. So apart from that, I also this textbook is giving us a kind of analogy. The analogy is that you should think of DNA as long sentences of 4 letters. Okay, so all the meaningful words, for example, when you make a sentence, Ada is a boy, that is DNA. So Ada is a gene because it is combination of this word. Ada is a gene, is a gene, a is a gene. So genes are meaningful words in the sequence, in the sentence. Then the whole genome, when you combine the sentences together, sentence 1 to the last sentence, when you combine them together, they are going to make a complete textbook. That complete textbook is a genome. So what I'm saying in essence is, even if you don't know what a DNA is, you should know that a DNA is a long sentence. The gene, which I said combination, that is segment of DNA, are known as the gene. Okay, so then when you now bring all these genes are just like sentences. So, and if you want to write a textbook, you are going to combine different types of sentences together before you have a whole textbook. So the whole textbook, as you can imagine it, is serving as the genome, serving as the genome. So scientists use computers because no human could read billions of letters by hand. So I have told you that it will take more than 95 years without sleeping for us to be able to read the human genome sequences. So the key terms and terminology in that is that you should know at this level that adenine, thymine, guanine, cytosine, all these are being abbreviated as ATGC. Then you should know that a DNA is a section of DNA. A genome is a complete DNA sequence of any organism. Then DNA sequences is like when you are reading and recording the order of bases in the DNA. So when you sequence, when you want to know all the alphabets of a textbook, That is when you will be talking about sequencing. So if I'm talking about sequencing, I'm really talking about how can you bring all the alphabets that make the whole textbook together. So what I'm saying in essence is, if you have a textbook, and that textbook is more than containing more than you start counting the alphabets that make the whole textbook, that means you are trying to do sequencing. Wanting to know the amount or the number of the alphabets in a textbook is, you are trying to know or sequence. So then Human Genome Project, which started from international project from 1990 down to the year 2003. So that was when the first Human Genome Project was reported. Now, under normal circumstance, you should see DNA sequences for the first time. How will you be able to see DNA sequence? So the learning objective is to move through the NCBI in your browser. At this level, you know very well how to get to NCBI, and you know the full meaning of NCBI. So what you are going to do is to search for Escherichia coli. Not only Escherichia coli, you can also search for any other um, organism in NCBI. And what I want you to pay attention to is, you pay attention to the formats. You pay attention to the formats. When you pay attention to the formats of reporting all this. So just like what I was saying, I'm going to give you when and when to do that. So then when you are now searching for any organism in the genome, I said you should pay attention to the formats, annotated formats. You should pay attention to it. Where do you have the headings? Where do you have the body? Where is the accession number? You should be able to know and identify all those. Then apart from that, in the whole sequence, you have to identify that all will be given into ATGC, that is in form of their alphabet. And that is exactly what makes the whole scenario very, very interesting. That's what make it so interesting, because we just discovered that all organisms, without any exception, they are already being synchronized or being synthesized into alphabet, that is DNA. Then don't forget, I have actually mentioned and I've taught you very well in this class about what is called the central dogma theory, which I said it is a flow of genetic information from the DNA down to RNA, then to amino acids. From DNA to RNA, you talk of a process that is called transcription. And from RNA to protein is going to undergo a process that is called translation. So I want you to know and be following me as we are going on. Then apart from that, you are supposed to write a very short description of what you have found. Part of the description of the sequences you are going to found is the format. I want you to take note of all these so that by the time you are going to NCBI or any database, you will be able to know what you are expected to do, so to say. Then, as a point of emphasis, I want you to know, once you get, please take note, once you get any sequence from any database, the first thing you should do is to clean that data. I'm going to give you the website that the first thing you will do is to do what? To clean the data. Why do you need to clean the data? There are something we call redundancy. It is machine that do what is called sequencing. And as you know from the law of physics or your elementary physics, that there is no machine that is 100% efficient. So that is, there are some certain errors during the process of generating this alphabet. There are possible mismatches, there are possible redundancy, there are possible omission, and there are also possible kind of a duplication. That's the reason why once you get your sequence in any form, will it be in RNA form, in DNA form, or in amino acid, the first thing you need to do is to clean that data. Then also, I have also told you in this class that it is very, very easier for you, and it's very, very advisable for you to actually work with amino acid, that is protein, because one amino acid, three nucleotide bases will make one amino acid. So what we are saying is that if you have 300 nucleotide bases, so by the time you now generate it into amino acid, you are going to have that 300 divided by 3. That is going to be amino acid, 100 amino acid letters. And it is advisable and very, very possible that in bioinformatics, the amino acid you work with is a single letter code, so which you should know. Now, the question now is, what is NCBI? The full meaning of NCBI is a National Center for Biotechnology that you can get at ncbi.nlm.nih.gov. So this is the world largest, please underline that, is the world largest free biology library. You don't need to pay any subscription before you get to this library. And in this library, it contains, when you get to library, you have a lot of different types of textbooks, okay? And don't forget that I have linking that textbook to the genome of different organisms. So when you get to the world largest free biology library, which is NCBI, so you don't need, no account is needed before you be able to browse. It's not going to say, oh, enter your Gmail or enter your email before you be able to have access to it. It is free. But if it is free, what are those information that you can find in that library? Number one, you are going to find DNA sequences from hundreds of thousands of organisms. So you see nucleotides, different nucleotides. Then you can also get to certain level, but that is the only one. Now, the laboratory steps that you have to take. Number one, you open NCBI in your browser. Then the next one is you search for Escherichia coli. If you are searching for another, you can also search for another organism. Instead of Escherichia coli, you search for your own. Okay, then you click on genome. Step 4, you open a result and find the sequence section. Open a result and find the sequence section. Then step 5, copy any 20 consecutive letters in your notebook. So that is, when you have, don't forget that each organism is made up of thousands, hundreds of amino acids. That is, these letters intertwined together to form a genome or a kind of sequence for that organism. So what you should do is just to copy the first 20 into your notebook by writing it down. You write the first 20, write it down. The next one is that you will count how many of each base. You will record how many A do you have in the 20, how many T do you have, how many G do you have, how many C do you have. So you can even copy more than 20. You can copy as much or as small as you might want to study. And don't forget that if you copy, irrespective of the number of number of nucleotide sequence that you copy, that would determine. If you want to know, that is the essence of what is called blasting. That is the essence of what is called blasting. So don't forget that blast is like you have the whole organism. You have the whole organism and you just pick some of the sequences from the whole genome, and you want to know where it is coming from, or you want to know which organism has that set of sequences. Then apart from the organism that has that sequences, you can also do what is called blasting by making some what you call comparison, alignment. You bring them together. I've told you that alignment means that you are bringing different substance or different sequences. You are bringing them side by side. If there are only two, you call it pairwise because it's dealing with two. You are bringing them together pairwise. And if it is multiple, that means the sequences you have more than two. So you want to compare it. That is multiple sequence alignment tool. So we will also see that. Now, the analogy from this presentation is that NCBI is a massive public library. But instead of you to find books in the library, what you find is DNA sequences of almost every living thing ever studied. And just like a library, it is completely free. I've mentioned that in this library, instead of you to find books books in geography, find books in biology, you have different segments whereby you say this one is a catalog for science, this one is a catalog for social sciences. So in the library of NCBI, what you found there is nothing but DNA sequences. And the DNA sequences that you found in this library is from different living organisms. It's not only on microorganisms alone. You also found that from plants, from or any living organism that you can think of that has ever been studied, and it is completely free. Then one other thing that I want you to know is that the format in which you should work with is called faster formats. Faster formats. So when we start working on this in the practical class, you will see that the format we are going to be retrieving, are going to be getting, is going to be in a faster format. And the faster format, we always start with greater than letter. Greater than, you have to take note of that. Greater than letter, and that is found in where you have the greater than letter is called the description line. description line. That's where you find the greater than sign. Then another thing is a browser-based tool. Browser-based tools like NCBI that you don't need to install them. One of the limitation that you might have as a phone user, as an Android user, without working with a laptop is that you can only have access to web-based tool. Those ones that are free, that you don't need to download. There are some databases that you will have to download them first. For example, if you want to plot phylogenetic tree, you will download you will download a software that is called Mega. That is called what? Mega. And like that and like that. So, but you should know that in NCBI, so just like I said, that in as a phone user, you have limitation to what you can do in bioinformatics. And that is why I said normally what you should do is to do bioinformatics on computer, that is on laptop or desktop. You can do it in that way. So when you say browser-based tool, these are tools like bioinformatics tool that you can only access them when you are online, when you have access to browse. So with no installation required. Okay. Um Now you are going to do the test. The test is already there. That's going to be test one. I'm already displaying it. So please take up your paper and do the test. Take up the paper and do the test. Please, just like you have it in the by now, you already have the okay. You have the question and you know the solution now. The second role play activities that you are to do is step into the science. So the instruction is you are to work with a partner or small group of 2 to 3. You are to read your role card very carefully, then carry out the activities as described. Your lecturer will give the signal to begin and we observe each group. Okay. This is expected to last for only 8 minutes. Students, so these are the how do they call this? These are going to be the actors or the role players. Okay, they are going to have students. Somebody is going to act as a student, another person is going to act as a aunt or uncle. So you have just returned from your first bioinformatics lecture. That is the assumption, the playing of that student. You have just returned home from your first bioinformatics lecture. Your aunt asks you what you are studying. So you have to explain to your aunt or your uncle the meaning of DNA, genes, and why computers are used in biology. So you have to use everyday language so that she will understand. So the task is for you to be able to demonstrate your knowledge about DNA, your knowledge about gene, your knowledge about genome, your knowledge about NCBI, why computers are used in biology, then some basic terms and terminologies like the Human Genome Project. So as for you to be able to break it down. For example, if somebody now asks you to everything you have been learning in bioinformatics, how can you explain it to a layman, somebody who is not even in science? And that is the major and the main objective of you learning anything at all. If you are learning anything, you don't need to cram. You don't need to cram. What you just need is to have a proper understanding. You should be able to imagine it. You should be able to dramatize whatever you have. And that's the reason why, as a student of bioinformatics, I have told you there are 4, at least you should have knowledge, very good knowledge about 4 AI tools. Number 1, you should know how to use ChatGPT very, very well. Very, very essential for you. The second one is Claude.ai. Claude AI. The third one is Gemini. Go and learn it. Gemini. Then the fourth one is Perplexity AI. So all these are part of what you should know as a student, not only as bioinformatics, as somebody who is a student in this 21st century that we call the digital era, where you have to put everything into being on your phone. Don't forget, all these genome, all these sequences that we are talking about, that alphabet of life. Don't forget that when you pick up an organism A that is causing problem, maybe somebody is very sick, and as a microbiologist, but you don't forget that you are not ordinary microbiologist. You are a biologist that understands life to the molecular level, life to the level of alphabet. So what you should do is collect the person's sample. You want to know what is the problem, the organism, the microorganism that is responsible for that problem. Okay, you go at those days. In those days, they would just say, "Oh, they diagnose diseases by only their symptoms. They will say, how are you feeling? I'm feeling headache. Okay. Your body temperature, they say, is above 37 degrees. The next thing is what other symptoms? Vomiting, diarrhea, and all this one. All these are just in those days. All those kind of diagnoses are in those days. For example, you can see that it is very, very difficult for somebody to diagnose malaria that is caused by a parasite, which is Plasmodium disease, and typhoid fever that is caused by a bacteria. Parasite that is protozoa is quite different from bacteria. Now, if you are to rely on the symptoms and the preliminary diagnosis by, okay, what symptoms are you? So you will find it very difficult to be able to differentiate the fever that is being caused by Plasmodium and that one that is caused by um, bacteria. And don't forget, once you misdiagnose, you are going to mistreat. Definitely. So gone were those days. Nowadays is when you are to diagnose and diagnose without any mistake. And the only way you can do that now as a biologist is to be able to diagnose to the DNA level, to the alphabet level. That is the sequencing. So you collect the person's sample, you go to the laboratory, go and culture. We call it wet lab. Go and culture, go and isolate. By the time you isolate, you grow it on media, different types of media. Apart from that media, you proceed to do what is called biochemical tests. Biochemical tests will only narrow it down to certain level. In fact, sometimes, if let me just tell you, I told you that we submitted a sequence in NCBI, maybe year 2021, that was 2021. Everything, all the preliminary investigation up to the biochemical level, we thought the isolate was E. coli and Pseudomonas. Everything we did. In fact, all symptoms, when we check through all the features and characteristics, that was what we thought. Until we now sent it out for sequencing. When we sent it out for sequencing, we now discovered that we have a very two different. In fact, what we got at the end of the day, we were talking about, we were thinking it was gram negative, but when the result came out, it was gram positive. So to actually tell you the reason why you should understand, the reason why you should understand that understand to the certain level of being a digital microbiologist. So what you should do, your aunty, you are a market, the aunty you are going to talk about, you are a market trader who study only secondary school to only secondary school level. So to do only secondary school level doesn't know anything about DNA, doesn't know anything about genome, nothing like sequencing, nothing like NCBI. It doesn't have any knowledge about that. Ask at least 3 genuine questions, such as, what exactly is DNA? As an auntie, you ask that student that has just learned bioinformatics lecture, ask the person, what is DNA? What do you mean by DNA? The first thing you do, tell the person the full meaning of DNA. What does DNA do? So what does why does a computer need to read the DNA? Why do you need to read? Then what does this have to do with sickness? So as somebody who is just a bioinformatician, how will you explain everything, all your knowledge that you know about DNA, why is computer so important, and what essence does the application of bioinformatics or DNA in disease diagnosis. So the scenario, you are at a family dinner. Just imagine that you are at a family dinner. Your auntie has just heard the word bioinformatics from your parents and is so curious, but very, very skeptical, very, very curious. You know what they call curiosity? Curiosity means you are trying to understand what you don't understand that is too difficult for you, but you are skeptical. Skeptical means that maybe you could be able to explain it well, or maybe you will not be able to explain it well. So until you are able to convince your auntie before the skepticism, we know very well that um, you are a very good student. So she thinks real biology is done only in the laboratory, not on computer. So you have to convince your auntie that auntie, although during your own secondary school, during your own time, you also said you read biology. You also said maybe even a professor during their own time, what they did was to strain their eyes by the use of microscope. What they knew then was um, uh, what they knew then was too different from what you are doing now. What we are doing now is that we are migrating fully. We are moving away from classroom where we will be talking about in chalks and in marker. We'll just be writing, drawing on the chalkboard, explaining difficult terms, explaining difficult scenario. But nowadays, our biology, just like in the introductory uh, lecture I told you that biology is now being done and being practiced. You can start it from the computer and you can also end it from computer. COVID-19, one of the one of the secrets why COVID-19 we were able to get vaccine was because immediately 24 hours after the announcement of COVID-19, those genomics at Ede, they were able to sequence the whole genome of COVID-19. And once you are able to sequence it already, you already have, because when you have information, correct information about your enemy, you'll be able to overcome your enemy. Is that not true? So because when the fight is just like the just like the biblical this thing that says we wrestle not against flesh and blood. So what we are saying in essence is just that we are not fighting against ordinary microorganisms. We are fighting their molecules. We are fighting their weapon. If we do not understand the weapon in molecular terms, the microorganisms, they are going to win and win always. And that's why part of their winning is to render our drugs, render our antibiotics, render them so useless to the level that it will no longer be able to kill them. So many people in those days, whenever there is any outbreak, people will just say, oh, don't for just go and take chloramphenicol. People will just try to recommend any antibiotics to them. But nowadays, those antibiotics are no longer useful. They are no longer effective against all those ones. And the reason is just that we have trained our enemy. We have given them our secrets. We only allow them to know about our secrets, and that's the reason why the enemy was able to go and prepare, was able to go and make sure that any other time that we make use of that antibiotics again, they will render it so useless. And that's the reason why the success of a biologist today is no longer restricted to the amount of wet lab. Huh? It's no longer restricted or limited. I mean, it's no longer restricted or dependent on the number of plates that you are going to read per day. In those days, I remember when I was working Sometimes I remember I was working microscopy, I think for more than 3 months, just working on slide, reading the slide, I would document and do every other thing. Then, but now I don't need those any longer. In fact, if you know some AI tools nowadays, what you just need to do is just to supply your slides to that, just snap it, screenshot it. There are some websites that when you even put them there, ask, use that AI tool to to identify and predict those organisms for you. They will be able to do the normal precision of it. So what we are saying in essence is your success as a biologist. And don't forget that biology is the basis for medicine, is the foundation for medicine. So as a biologist, if you don't understand how to use computers to be able to make complex decisions, be able to do a lot of analysis under normal circumstance, you just think that everything you will be saying, people will not even value you at all. And that's the reason why your being part of this course, it is expected that you should start thinking better than those in the past. Then I need to also tell you this. I've been teaching bioinformatics for almost 5, 6, 7 years ago. And let me just tell you that every year we normally have it in different form, more advanced. The bioinformatics I taught last year wasn't as advanced as this. Advanced in the sense that I remember that time I just joined all of them together, HND2, packed them together, microbiology. I will have a very bogus class, and everybody will come, they will make noise, they think, they learn, they do that, and they will go back. But now we have already, one way or the other, deployed a kind of cohort group to be able to understand better and have good access to that. That time, I know how much we normally used to spend on fuel that time. Not to even talk. In fact, there was not even any internet access that time. So everybody will just come, people will be complaining, their data is this and that. But as I told you, as a bioinformatician, your secrets, your secret is on computer. Your secret is on the use of internet. And gone were those days. Before you be able to even work on computer, in those days, you need to say, oh, you need to understand some technical terms. They will teach you, they will say, okay, you need to go and learn 12-month course before you be able to even work on Microsoft Excel. But nowadays, once you are familiar with the keypad in your phone, on your phone, normally you should be able to navigate and work effectively on the computer. So what I'm trying to say is just that I want you to have a very good understanding about what bioinformatics is all about. And you should be able to ask very useful questions and so to say. Okay. Then another one is a group assignment. The group assignment here is group 1. We are going to work on human genome project, be able to understand it. Then group 2, we work with titled DNA in everyday life. DNA in everyday life. Okay. The group that we work on human genome project, what are you expected to do? You are expected to talk about when human genome project started and when it was completed. How many countries were involved? What was the cost? What are the 2 things scientists discover because of it? Then you are to present a kind of talk, like 5-minute talk. You should be able to say something about human genome project. Then the DNA in everyday life, you will find 3 real examples from Nigerian life. Nigerian life where DNA knowledge is used. For example, in paternity tests. Determination of who is the right father of a baby, crime investigation, disease diagnosis. For each, explain what DNA tells scientists that other methods or not actually test. Then group 3, you have to compare what? Genome sizes. You go to NCBI, you look up and record genome sizes. That of Escherichia coli, how many base pair? That of Plasmodium falciparum, how many base pairs? And Homo sapiens, that is human, how many? Then a plant, any plant of your choice, you can pick any plant, how many base pair do they have? Then apart from that, you can also pick a virus of your choice. Then you will now create a simple table ranking them from smallest to the highest. So it is this table that you are expected to submit. Then the group 4 will talk about DNA basis creative projects. You have to create a creative visual project or a written aid to help a beginner to remember the 4 nucleotide bases of a DNA. So what can you do? What is the learning secrets that you can give so that whenever anybody wakes you up at any point in time, you'll be able to remember the 4 nucleotide bases of a DNA. So this could be in form of a diagram. You could write a poem in form of it. So what you can do if you want to write a poem is that you go to ChatGPT, tell ChatGPT to compose a poem that will help you remember nucleotide bases. It's going to write it for you. You can even ask it to tell you a story that will also make you to remember the nucleotide bases. Honestly speaking, just like I said, going by those days that you should start memorizing, cramming things. But nowadays, you should make sure that all these are then the best submission will be displayed in the classroom, okay? Then group 5 we work on NCBI exploration reports. You are to explore the NCBI websites as a group. Then you have to identify 4 different types of databases available on the site. How can you search for a Nigerian organism? That is, organisms that are only from Nigeria. How can you search for it? That is what group 5 will do. Then what the gene bank section shows. So you have to write a short guided tour for a classmate who has not visited the site. So, and this you have to write just 1 page written that you will tag it or title it NCBI guided tour. That is the group assignment. Then apart from that, that will now help us to move to week 3, and that is microbes and data. How bacteria and viruses leave their digital fingerprint. You have studied microbiology or microorganism in microbiology. Under normal circumstance, microbiology is the study of microorganisms, and these microorganisms are divided into 4: bacteria, viruses, and fungi. Okay, bacteria, viruses, parasites, and fungi. So in this week, we are going to look at these 4 characteristics, these 4 group of microorganisms. Then how can we, what can we make of their data? Now, at the end of this week lecture, you should be able to explain a microbial DNA fingerprint. If you say microbial DNA, what do we mean? Then you should be able to describe how scientists differentiate bacterial strain using DNA. Explain how Lassa fever is tracked using genomics. Then you should be able to connect um, DNA fingerprints to a real this. So what we are going to do, just like I said now, is that go and focus on the group, on the presentation, and I am going to, that's where you will do the next set of questions. So you are going to do 10 questions, and as soon as I'm writing them, you are expected to uh, respond in form of a reply under that. Okay, so don't forget to only 6 people showed love on this. Only 6. In fact, some of you have even logged out before. Hmm. Cool. Hmm. Not sure. Oh, yeah, 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 yeah. Okay, do you know what? Don't just concentrate on the comment section. Don't write that one down. The one you are going to write, I will tell you. The one you are going to write, I will tell you. So only reply to it on comment section so that you do not get distracted on the comment section. Make sure you reply, you write the response as a reply to that question. Okay. That explain genes, DNA, genome, and bioinformatics to a layman. How can you use bioinformatics to diagnose a new disease outbreak in your area? Okay. If you cannot write a comment, that means why? Have you subscribed? When did you subscribe? It's either you have not subscribed or maybe certain responses in the past you have not been responding to them. You have not subscribed. That is it. If you have not subscribed, you won't be able to comment. I have told you times with that number. Is this your first time of attending this lecture? Did you attend?
So to that particular question, how to reply it? Not really.
Why?